Okay, I'm here with Nick Spitzer, who's Distinguished Professor of Neurobiology, co-director of the Kavli Institute for Brain and Mind, and founding editor-in-chief of brainfacts.org. Okay, starting in general terms, what is the elevator pitch version of explaining your research?
I keep it brief, I keep it simple. I say that I'm a neurobiologist, I study the brain, and in particular, I study the way the brain changes in response to experience. And then if they want to know a little more, I tell them that these kinds of changes, of course, are what are involved in learning and memory. And typically, everyone is interested in, if not learning, certainly having a good memory. So that usually is something that resonates with people. Now, if they want to know more, I'm looking at them, trying to judge from their response whether they've about had it or whether they are eager for perhaps a few more details. I can go further. I can tell them that we have discovered a new way in which the brain changes in response to the environment and experience. And it involves the way nerve cells talk to one another. There are billions and billions of neurons in the brain. The latest count would say 86 billion, big number. And these nerve cells have to communicate with one another. They do this by releasing a chemical that interacts with the next nerve cell, the nerve cell next to it. And our discovery was that the chemical can actually change, can flip, can switch. So that's been very exciting. And if they're still interested, I go on to tell them about the change in the sign of this signal, going from excitation to inhibition or going from inhibition to excitation, and how this then leads to changes in the animal's behavior. We can even go on to talk about how that may be implicated in the origin of different kinds of mental disorders.
And then conceptually, what are the most difficult aspects in explaining your research to the public?
The thing that people, the layperson typically doesn't understand is this process of synaptic connections. Nerve cells come very close to one another. They don't actually fuse with one another, but they communicate with one another at these sites of proximity where a chemical is released from one neuron and diffuses a short distance. It sounds like an engineering nightmare, but they bind to receptors on the next cell. And that process is something that the undergraduates in my courses here get, of course, early on and pick up right away. But the lay public doesn't understand that. And so I have to find some way to introduce that communication process. Then the idea that the chemicals that are used in that process can change seems certainly plausible. We get, hopefully at some point with some of the audience, to the point of understanding that when the chemical changes, very often the sign of this connection from excitatory to inhibitory switches. This is, I think, for me and for most of us working on this process of transmitter switching, this is really the bee's knees. This is really very exciting and could account for the behavioral changes that accompany this transmitter switching.
And what techniques or strategies do you find helpful in explaining your research to the public?
I find it very helpful when communicating our research to be sure that I've judged my audience appropriately. For an audience, perhaps a few people who have a limited interest, I will try to keep my communication chunks brief so that I don't saturate their interest, so that they can follow them and we can have some give and take, back and forth, more of a dialogue than a monologue. I find that usually keeps them engaged better. For a longer presentation format, I find it very helpful to tell stories. People love to hear stories. They should be, whenever possible, personal stories, because if you're sharing something personal, this, I think, has a greater capture rate in terms of the audience's interest. When I can, I try to introduce humor. Being funny is an incredible asset in any presentation. The thing I have great difficulty with is that I'm not a really good stand-up comedian. There'll be days when I'll be giving lectures here on the campus to undergraduates, and I'm just the funniest guy they've ever heard. And I don't know what it is I'm doing differently. I wish I could capture it and plug it in every day. But of course, that's real magic in terms of getting a story across. So if one can think of ways to introduce humor, it's terribly valuable.
What metaphors or analogies have you found useful in explaining your research?
After I give my initial one-sentence lead-in there, I go on to say that I'm very interested in the particular mechanism in the brain by which cells change their ability to communicate with one another. We know that the 86 billion neurons in the brain have to talk to one another. That's how our brain works. And we have discovered a new way that they talk to one another. And then, of course, if they say, well, gee, what is that new way? Then I say, well, we found that the neurons change the chemicals that they use to speak to one another. It's kind of like switching from English to Spanish, or maybe from Spanish to Chinese. So they're changing the language that they use to talk to one another. And I'm careful to titrate my audience because people have a limit to how much they're willing to engage. And it's probably quite important just to try to reach that limit and then maybe back off a little bit.
How do you come up with these metaphors and analogies? How do they arise in your research?
One problem that has come up in explaining our work to both to the public and to other scientific colleagues has been that people are much more familiar here at the start of the 21st century with genetic origins of disease. The molecular biological revolution that put molecular biology on the map for everyone in a research lab allowed the identification of gene mutations that could underlie a variety of neurological problems. It's the process of identifying the activity-dependent, experience-dependent, environmentally dependent disorders has taken a longer period of time. I believe it's now coming into fruition, where people are now beginning to appreciate, yes, this is a reality. But helping people understand that in addition to the important origins that arise genetically, that there are these activity-dependent origins has been an effort.
Your research uses lots of different tools and techniques from different fields of biology, but the public's often not in the know in terms of what happens in there. So how do you let the public and even journalists into this black box, and how do you explain that to them?
Yes. I think it's possible, given the interest, perhaps, of a journalist or someone who wants to try to explain something, perhaps in writing or in a video, to make it accessible to an audience, to explain why at any particular juncture in the experiment one turned from one technique to another technique, and why this technique at this time, with the maturity of the answer to the question that we're trying to develop, was really the best technique to employ to advance the understanding to the next level. Typically, addressing a scientific problem is a little like peeling an onion. You start at the outside and you see the problem in its sort of large scale, but you want to dig deeper, and so you take off the outer layers of the onion, and then you get a little understanding of how those inner layers are organized, and then after that's been appreciated, you can peel off some further layers, and finally, you're down just at the very core of the onion. And I think the public would probably be interested in knowing how one takes these steps. From the outside, it must look fairly opaque. How did the scientists, how did we decide to take this particular path, this particular route? We don't typically say much about that, and I think it could be a part of the richness of the story, coming back to the importance of telling a story, that would keep the audience more fully engaged.
Your research is aimed at the basic biological processes, but there's lots of implications for human health. How does that influence the way you explain to the public about that process of discovery?
I try to gear my discussion with a friend or a colleague or a person I might meet on the campus or across the country to what I perceive to be their interests. I can talk about the significance at a basic level of what we are doing. Very often, people appropriately are very interested in neurological and psychiatric problems. They have a relative or a friend who suffers from a disorder. And so I tell them in those cases how I think the sort of brain plasticity we are studying, how that can be related to the generation of these disorders, and how understanding the process of transmitter switching may help us provide treatments for those problems. That is often a story that people are very interested in hearing because of the connections that they have with people who have some of these problems.
What advice would you give to someone starting out in either biology or another field, just starting out in terms of science communications and how to help them better communicate to the public?
Keep it brief, keep it succinct, be enthusiastic. People like to hear about work that the individual is very positive and excited about, and don't oversell the story. Keep it real, but try to follow up with individuals to the extent of the depth of their interest. This will be incredibly valuable. The public needs to know more about how science is done and what the values of scientific research are. And everything that we can do in that regard should get top priority.
And finally, is there anything else you'd like to add?
I have been involved for a long time with the Society for Neuroscience. It's a big scientific society, 40,000 members, has a big convention here in San Diego every third year typically. And the Society for Neuroscience has long been devoted to trying to communicate science to the public, neuroscience to the public. And of course, in the early days, back in the 20th century, print materials were the order of the day. Now in the 21st century, with the availability of the internet, the availability of the richness of images and videos, this represents a new and very important dimension with which to communicate with the public. And I think that this is going to continue to be increasingly the case. That, and I think it's going to be important to take advantage of these media, because what I can do as one person on the campus, admittedly, lecturing to thousands and tens of thousands of undergraduates, is really quite modest in terms of what we would like to hope to do in terms of reaching the global population and communicating the excitement and the importance and the significance of of neuroscience and neurobiology.